Omega-3 fatty acids have long held a favorable reputation in the health and wellness world, particularly for their cardiovascular and anti-inflammatory properties. Rich in EPA, eicosapentaenoic acid, and DHA, docosahexaenoic acid, omega-3s are celebrated for reducing triglycerides, improving endothelial function, and supporting brain health. Naturally, this favorable profile led researchers to investigate their potential role in slowing biological aging, especially by examining their impact on telomeres, the protective DNA sequences at the ends of chromosomes. Telomeres shorten over time as cells divide and are increasingly recognized as key biomarkers of biological age. However, despite the theoretical promise of omega-3s in cellular protection, clinical trials have found inconsistent and often underwhelming results when it comes to telomere preservation. This raises an important question. Why haven't omega-3s delivered the same telomere protective effects seen with vitamin D? To begin, it's essential to understand that telomere length is influenced by a broad array of cellular processes, including oxidative stress, inflammation, DNA repair capacity, and enzymatic activity via telomerase. While omega-3s do provide anti-inflammatory benefits, they may not target these pathways as directly or as robustly as vitamin D. For example, vitamin D has been shown to influence telomerase expression, the enzyme responsible for maintaining telomere length in certain cells. This direct genomic action allows vitamin D to influence the rate at which telomeres shorten, a mechanism that omega-3s, despite their broad anti-inflammatory properties, do not appear to engage as effectively. Moreover, omega-3s function largely by modifying lipid-based signaling pathways and cell membrane dynamics, which, while beneficial to cardiovascular and neurological health, may not strongly affect the genetic and chromosomal maintenance systems that control telomere integrity. In contrast, vitamin D functions more like a hormone, entering the cell nucleus and binding to vitamin D receptors, VDRs, to regulate the expression of hundreds of genes, many of which are involved in DNA stability, immune regulation, and cellular aging. This genomic reach gives vitamin D a broader and potentially more profound impact on cellular aging markers like telomere length. Another factor could be the timing and dosage used in many omega-3 trials. While omega-3s are effective in reducing inflammation over long periods, telomere shortening is a slow process, and subtle protective effects may take years to become apparent. Some trials may not be long enough or may not use high enough dosages to reveal these effects. Moreover, not all omega-3 supplements are created equal. Differences in purity, EPA/DHA ratio, and the oxidative stability of the oil may influence the outcomes. If the omega-3s used in studies were oxidized or poorly absorbed, their impact on telomere biology might be muted, despite theoretical benefits. It's also worth noting that baseline inflammation levels and oxidative stress differ among individuals. In people with very high levels of inflammation, such as those with metabolic syndrome or autoimmune conditions, omega-3s may offer more pronounced benefits. However, in relatively healthy populations, their anti-inflammatory effects might not be strong enough to translate into measurable telomere protection, especially in comparison to nutrients like vitamin D, which act on more specific genetic targets related to cell aging. Importantly, the limitations of omega-3s in preserving telomere length do not diminish their overall value in a health-promoting lifestyle. They remain powerful allies for reducing cardiovascular risk, supporting mental health, and maintaining cognitive function. But when it comes to anti-aging strategies aimed specifically at genomic stability and telomere preservation, they may need to be paired with other nutrients or lifestyle interventions to create a more effective cellular defense. This contrast between omega-3 and vitamin D also underscores the importance of precision in nutritional science. Not all nutrients affect aging in the same way or through the same mechanisms. Vitamin D may stand out in telomere biology because of its hormonal nature and ability to regulate gene expression directly related to cell life cycles. Omega-3s, on the other hand, serve more as systemic moderators of inflammation and lipid metabolism, critical for whole body health, but perhaps too indirect to serve as a frontline defense against telomere erosion. In summary, while omega-3 fatty acids are invaluable components of a healthy diet and offer broad physiological benefits, Their role in preserving telomere length and directly slowing biological aging appears limited when compared to the more targeted, gene-regulating effects of vitamin D. Rather than viewing this as a failure of omega-3s, it may be more accurate to see it as a call to adopt a more comprehensive strategy, one that combines nutrients with complementary functions to optimize cellular longevity and overall well-being.